Hi and welcome to Night Science Stories. Pressure more than 1,000 times higher than at the surface, water temperature close to the freezing point, and darkness complete from horizon to horizon. These are the baseline conditions at the deepest place on Earth. Equipment fails here if a single gasket is chosen poorly. Metals creak. Even light must be carried. Yet life is present. It is not just surviving. It is using these conditions as its normal state. The surprising finds in the trench do not begin with large creatures. They begin with microorganisms that treat crushing pressure as ordinary weather. The trench hosts communities of bacteria and of archaea that metabolize without sunlight. Instead of photosynthesis, they rely on chemistry. Some oxidize ammonia into nitrate through a pathway that works best at low temperature. Some reduce sulfate to sulfide using organic carbon that falls from far above. Others use hydrogen or methane when those molecules seep from the crust. They are pisophiles, a precise term that means pressure-loving. If you remove the pressure, many of them die. Adaptations appear at every level of their biology. Cell membranes in these microbes are unusually flexible because they contain more unsaturated fatty acids. That fluidity counters the tendency of pressure to stiffen the membrane. Proteins inside the cells are supported by small molecules called osmolytes that stabilize shape. Trimethylamine oxide, spoken slowly as trimethylamine oxide so that the words are clear for audio, is one such molecule. It prevents proteins from collapsing under pressure and also improves function at low temperature. Enzymes from these cells frequently show subtle changes in the amino acids at their cores, adjustments that keep the active sites working when pressure increases the density of water around them. In sediment cores taken from the bottom, scientists have recorded rates of respiration that are higher than predicted for such a cold and dark place. The explanation lies in the constant rain of organic matter down the water column and in the special structure of trench topography. The V-shaped profile of a trench collects particulate material that drifts sideways as currents bend around the slopes. It is a natural funnel. Microbes use this steady input like a slow conveyor belt of resources. In culture, strains of Moritella and of Colwellia, isolated from near the trench, have shown optimal growth at pressures that would crush an unprotected instrument. Their names appear in the literature as signals of a lifestyle that does not translate to the surface. Viruses are part of this invisible web. Bacteriophages, the viruses that infect bacteria, occur in the trench just as they do in other parts of the ocean. Their presence is not a footnote. Viral lysis releases nutrients back to the dissolved pool and changes the flow of carbon. The turnover is one reason the trench does not become an inert storage locker of organic debris. Processes run slowly, but they run without pause. Among the most arresting organisms in video frames from the bottom are giant single cells called xenophyophores. Each one is a foraminiferan that builds a delicate test or shell by gluing together fine particles from the sediment. Some individuals span several centimeters across and hold thin internal chambers that look like lace under a microscope. They are not colonies. They are single cells that reach sizes normally associated with small animals. Where calcium carbonate dissolves under pressure and in water rich in carbon dioxide, these foraminifera replace mineral tests with organic ones. That is a direct response to the chemistry of deep water. The xenophyophores form carpets that trap other particles, increasing the surface area available for bacteria and small animals. In effect, a single cell engineers a small habitat that benefits a miniature ecosystem. Another set of single-celled actors are allegromyid foraminifera. They build flexible organic tests and occupy pore spaces in the sediment where oxygen has been lowered by microbial activity. Many of these foraminifera house symbiotic bacteria that help process nitrogen. The combination of foraminifera and symbiont acts like a slow filtration unit for the surface layer of the seafloor. Chemical surprises sit beside biological ones. Sediments from the deepest sites have yielded traces of industrial chemicals that were banned at the surface decades ago. Polychlorinated biphenyls and related compounds are present in amphipods that feed on detritus. These molecules did not dissolve away harmlessly. They rode particle after particle down to a place that many assumed would be free of our materials. The concentrations provide a quiet index of how persistent human pollution can be in the global ocean. Microbial genomes recovered from trench sediments tell an additional story. Genes for breaking down long-chain hydrocarbons appear across many taxa. Genes for tolerating heavy metals are present and expressed. Many sequences encode for chaperone proteins that refold enzymes after stress. Even ribosomes, the molecular machines that build proteins, show subtle modifications that appear to help translation under high pressure. If one asks what life has learned at 11,000 meters, one answer is that stress management is an everyday task. The microbial communities also govern rates of nitrogen fixation and of denitrification. Some convert nitrogen gas to bioavailable forms. Others send reactive nitrogen back to an inert state. In many cores, oxygen is consumed quickly within the top few centimeters, and then manganese and iron oxides take over as electron acceptors. That cascade defines which microbes can live where. When conditions favor sulfate reduction, hydrogen sulfide accumulates and feeds another group of organisms that can use it. Even without sunlight, the energy budget can support complex interactions and a high diversity of metabolisms. Large filamentous bacteria build mats near localized seeps where hydrogen-rich fluids percolate. These mats resemble the biofilms of shallow hot springs but grow at a temperature only a few degrees above freezing. Each filament is a chain of cells embedded in a sheath that helps channel nutrients and perhaps shares mechanical strength against the gentle but constant motion of the water column. Under a microscope, you can see sulfur granules shining inside the cells like tiny beads. The presence of such bacteria confirms that chemical energy from the crust occasionally reaches the trench floor and that microbes are ready to harvest it. Viruses again participate, this time with an effect on evolution. Some viruses carry genes that help their hosts use sulfur or nitrogen more efficiently. When such genes are expressed during infection, host metabolism accelerates. The virus benefits by creating more building blocks for its own replication. The host population benefits indirectly by acquiring new functions. Over long periods, this transfer of useful genes blurs the line between hosts and their parasites and speeds the innovation of metabolic pathways. There are also archaea that oxidize ammonia at very low concentrations. These organisms are so efficient that they maintain activity where an instrument barely reads a signal. The product of their work, Nitrite accumulates sufficiently to feed a second group that converts nitrite to nitrate. Together, they complete the first step in the nitrogen cycle under conditions that would stall many surface microbes. Laboratory cultures kept at pressures that mimic the trench reveal that the enzymes of these archaea bind ammonia more tightly and release energy more gently, an adaptation to conserve resources. Not everything in the microbial story points to endurance. Some microbes discovered in the trench are surprisingly fragile outside of their high-pressure environment. When researchers recover them using pressure-retaining samplers and then relieve the pressure too quickly, the cells rupture or their membranes leak. It is an immediate demonstration that we are the extreme ones, not they. The pressure that seems impossible to us is the comfort zone for them. The microorganisms provide the context for larger animals. What looks like an empty plain of mud is not empty at all. It is a slow farm managed by bacteria and grazed by small crustaceans. Near the middle of the food web are amphipods. The species Hirondelia gigas is one of the most studied. It swims in a jerky pattern close to the bottom and rushes to baited cameras in large numbers. Chemical analysis of its stomach suggests that it can digest cellulose from wood that filters down, a rare trait in the deep sea. The trick seems to involve enzymes that work under pressure and molecules that strip aluminum from sediment and bind it as a gel on the amphipod shell. The thin coating may protect the exoskeleton, which otherwise would be prone to dissolution because calcium carbonate is unstable in the high carbon dioxide deep water. Amphipods bring us to fish because they are the prey that supports the deepest vertebrates known. The record holders are snailfish. The species described from the trench is named for a naturalist who sailed during an early exploration of the Pacific. These fish live at depths greater than 8,000 meters, beyond the reach of most other vertebrates. They are not large. Their bodies are soft with minimal mineralization of bone and no heavy scales. The skull is partially cartilage. The muscles are pale and contain high levels of trimethylamine oxide, the same molecule used by microorganisms for protein stability. The concentration increases with depth. There seems to be an upper limit where the body would become so filled with these osmolytes that cells could not function. That limit may set a maximum depth for fish that is only a little deeper than where the Mariana snailfish actually lives. The sensory system of snailfish is tuned to a world without light. Eyes are small. Many individuals have reduced visual pigments. In compensation, the lateral line system that detects water movement is well developed. It allows them to hunt amphipods that swarm around patches of organic matter. Mouths are wide and teeth are small, reflecting a strategy of gulping rather than tearing. Stomachs have been found packed with amphipods, evidence that energy is collected in bursts when prey density permits and stored until the next burst. Reproduction in these fish appears to follow a schedule that respects slow growth in the cold. Eggs are large compared to body size, with thick shells that resist pressure. In the laboratory, related snailfish from shallower trenches show parental care. Whether the deepest species guard eggs is not yet confirmed, but the sizes of oocytes suggest a low number of high investment offspring. This is a common pattern in extreme environments where mortality from factors other than predation is high. Why do other fish not follow them deeper? The osmotic pressure imposed by stabilizing molecules seems to be the decisive limit. Theory and experiment agree that to keep proteins functional under higher pressure, a fish would need such a high concentration of stabilizers that water movement across membranes would become energetically prohibitive. In short, the chemistry that protects structure would break water balance. Rather than evolve impossible workarounds, fish stop at depths where the trade-off makes sense. That boundary is a natural experiment in physiology and evolution. Other unusual fishes visit the slopes of the trench. Grenadiers, also called rat-tails because of their shape, patrol the abyssal plains and the upper hadal zones. They are not trench specialists, but their presence is a constant in baited camera footage. Some eelpouts and cusk eels approach the edges of the trench. These groups use pressure-tolerant forms of hemoglobin and maintain buoyancy with lipids rather than with gas bladders that would collapse. None challenge the snailfish for depth records, but they help demonstrate that there is not a single recipe for life under pressure. Returning to the microbial foundation, an equal lesson emerges about the carbon cycle. Measurements show that microbial communities at the trench floor remineralize a significant fraction of the organic carbon that reaches them. The rest is buried. Over geologic time, that burial contributes to long-term carbon storage. Trenches act as temporary reservoirs because they collect particles faster than surrounding plains. Earthquakes occasionally disturb the deposits and drive turbidity currents that sweep material downslope into deeper basins. These events mix old and new layers and create lenses of unusually fine sediment where microbes respond with brief increases in activity. Observational technology shapes what we know. The crude bathyscaphe Trieste reached the deepest point in the year 1960. It returned a short report that included a flatfish sighting that later observers have questioned. A half-century later, a purpose-built submersible piloted by a filmmaker reached the same zone and stayed longer. It recorded amphipods, sea cucumbers, and jelly-like organisms. In the years since, free-falling landers equipped with cameras and chemical sensors have repeatedly visited the trench, confirming the presence of active communities. Pressure-retaining samplers now allow researchers to bring mud up while keeping pressure near in situ values. That step alone transformed our understanding of trench microbes, because many species simply did not survive earlier methods. An aspect that tends to be overlooked in popular summaries is the precise mineralogy of the sediments. With depth, calcium carbonate disappears, the clay fraction dominates. Volcanic ash from eruptions in the island arc contributes glass shards and altered minerals. These materials deliver trace elements that microbes can use as cofactors in enzymes. Manganese nodules, which are common on abyssal plains, are scarce here, probably because the steep slopes do not permit quiet growth over long times. In their place, concretions of barite and phosphate sometimes occur where microenvironments allow precipitation. Trace gas measurements reveal low but detectable levels of methane in some layers. Stable carbon isotope signatures suggest that the methane is a mix of thermogenic origin from deeper sediments and biological origin from acetoclastic pathways in the upper layers. Archaea that carry the genes for methane production are rare in counts but disproportionately active according to transcript data. Their role may be localized around pockets of organic, rich material. A surprising discovery in the past decade has been the presence of microplastics in trench sediments and in the tissues of amphipods. Particles in the size range of tens to hundreds of micrometers are common. Polymer types include polyethylene, polypropylene, and polyester. When researchers analyze the chemical signatures, they can match some particles to common textile fibers. The route to the bottom is simple and sobering. Fibers pass from wastewater into the sea, attach to organic matter, and sink. Amphipods ingest them along with detritus. The presence of microplastics in the deepest amphipods suggests that there is no level of the ocean uninfluenced by surface activity. Heavy metal concentrations reveal similar patterns. The metal element lead and the element mercury are present in small amounts in sediments, along with cadmium and others, likely carried by particles. In animal tissues, these metals are usually found at low levels because the local food web is short and there is little biomagnification compared with surface ecosystems. Even so, the record shows that the trench is a collector not just of particles, but of global history. From a physical point of view, the water column above the trench is a stratified archive. Nutrient-rich deep water flows slowly through the region as part of the global conveyor circulation. At shallow levels, the oxygen minimum zone can be pronounced, shaping the vertical distribution of bacteria and protists and altering the composition of organic matter that reaches the trench. Sinking particles change character as they fall. Lipids become oxidized. Proteins are broken into amino acids and then further into ammonia. Sugars are consumed quickly. By the time the material reaches the bottom, it is a selected diet to which the resident microbes are adapted. Research teams have also found filamentous fungi in sediment samples. These fungi tolerate pressure and likely colonize particulate organic matter rather than live free in the mud. Some produce enzymes that break down lignin, the tough polymer from plant cell walls that survives long transit from land. Their presence broadens the list of eukaryotes that participate in deep carbon processing and opens questions about spores and dispersal in the water column. Protists are diverse. Flagellates graze on bacteria and are in turn eaten by small animals. Ciliates with complex mouthparts navigate among grains. Tests constructed by agglutinating particles come in many forms and sizes. Some are delicate and collapse when retrieved without pressure control, which means they were undercounted in early surveys. With newer techniques, the diversity is higher than expected, approaching the values measured in shallower abyssal zones. The trench slope communities include small holothurians, commonly called sea cucumbers, that use tentacles to collect sediment for feeding. Some species are transparent and show internal organs in high-definition footage. Their slow motion resembles time stretching because there is no reference for scale in the absence of plants or other landmarks. For the animals, the feeding strategy is simple and effective. Ingest sediment, extract organic molecules and microbes, and pass the rest. Tracks on the bottom preserve their trails until currents erase them. Returning to fishes, there is evidence that the distribution of snailfish near the bottom of the trench is structured by temperature gradients and by prey abundance. In areas where amphipods concentrate, baited cameras record more fish and in shorter intervals. In warmer pockets where water from shallower layers intrudes, growth rates may be marginally faster. The differences are subtle. the unifying theme remains scarcity, punctuated by episodic abundance. Hearing in the deep is often overlooked. Sound travels far in water. Even in the trench, background noise from the surface storms propagates downward, although at reduced intensity. Some fishes use the lateral line primarily, but bones of the inner ear can also detect low-frequency pressure changes. That capacity may help them find prey swarms as amphipods respond to the fall of a large food item. One final layer of the microbial story involves spores and dormancy. Some species form resistant stages that endure long periods with little energy input. When a pulse of fresh organic matter arrives, enzymes activate and metabolism resumes. This ability to wait fits the patchy delivery of food to the trench. It also means that when sediments are disturbed by earthquakes, as they occasionally are, the community can reassemble quickly. Putting these elements together, the deepest place on Earth is not defined by absence. It is defined by a particular balance of chemistry, physics, and biology. Microorganisms exploit chemical gradients and adapt their molecular machinery to pressure and cold. Single-celled giants build structures that act as microhabitats. Amphipods process detritus and move energy up to vertebrates that live at physiological limits. Fishes exhibit a suite of anatomical adjustments that keep them within narrow bands where life remains possible for their lineage. Alongside this natural system, markers of human activity are present as chemicals and as microplastics, reminding us that the deep ocean is connected to our actions through pathways we do not always see. As research continues, new tools arrive. Autonomous landers now carry incubations that measure microbial activity in situ without ever raising the sample. Optical systems record changes in the microlayer above the sediment where chemical gradients are steepest. Genetic surveys recover entire communities from a spoonful of mud and link sequence data with microscopy. Pressure-tolerant cameras ride along in fleets to capture time series rather than snapshots. Each edition refines the picture. The story of what scientists have found in the trench can therefore be summarized in a calm list that remains open at the end. They have found specialized microbes that turn chemistry into slow energy without sunlight. They have found giant single cells that act as architects of micro-landscapes. They have found amphipods that digest wood, coat their shells with aluminum bound by chemistry, and carry pollutants that began as industrial products at the surface. They have found fishes at the predicted boundary of vertebrate endurance, with bodies tuned to cold and pressure. They have recorded viruses that shape ecology and evolution in silence. They have measured carbon burial and carbon processing rates that connect the trench to the climate story. They have documented the presence of plastics and metals, evidence that no depth is untouched by human activity. All of this knowledge serves the practical goals of ocean science. It guides the design of equipment that can operate reliably in the huddle zone. It informs our understanding of global biogeochemical cycles. It helps decide where protected areas would be most meaningful. It contributes to the long practice of exploration that does not romanticize the world, but treats it with patient attention. The deepest place does not change quickly. What changes is our ability to visit without disturbing and to listen without projecting our assumptions. In the trench, life is ordinary by its own standards. Microbes glide through reactions as if pressure were as unremarkable as weather. Fish swim where chemistry allows, not because they want to be extraordinary, but because that is where their lineage led. That quiet normalcy is the final finding. The extreme feels extreme only because we are not adapted to it. The inhabitants are not outliers in their own frame. They are the baseline of a world that happens to lie below 11,000 meters of water. Giant amphipods dominate many frames recorded at the bottom. They arrive within minutes after a baited lander touches the sediment. Numbers build quickly and then stabilize as the scent disperses. The behavior is consistent across deployments. Individuals patrol close to the seafloor, antennae forward, using chemical and mechanical cues to guide short bursts of swimming and walking. The body plan is standard for amphipods, but proportions differ at depth. The head and thorax are robust, the abdomen narrower, and the antennae long. The largest hadal forms reach lengths measured in tens of centimeters. Supergiant species from other trenches exceed 30 centimeters. At the deepest stations of the Mariana region, the typical size is smaller but still large compared with shallow relatives. Mass is carried as muscle and as lipid. The exoskeleton is relatively thin in carbonate because calcium carbonate becomes unstable in cold water that holds more dissolved carbon dioxide. Chitin and proteins take a larger role in structure. Trace elements from sediment contribute to surface chemistry in unusual ways. One species in particular, Hirondelia gigas, shows a surface coating rich in the metal element aluminum. The aluminum does not originate inside the animal. It is leached from clay minerals during digestion by organic acids produced in the gut. Once in contact with alkaline seawater, the aluminum forms a gel of aluminum hydroxide that settles on the exoskeleton. The layer is thin but consistent in specimens that live below the carbonate compensation depth. The practical result is protection. The gel slows chemical attack on the exoskeleton where carbonate is scarce. It also appears to reduce abrasion. This is an example of environmental chemistry repurposed as armor. Feeding biology explains the constant presence of amphipods around camera landers. They are opportunistic scavengers and effective processors of particulate organic matter. Laboratory assays show activity of enzymes that break down cellulose and hemicellulose. Wood that sinks from the surface becomes usable food. Lipid-rich material from dead animals is also consumed quickly. When a large food fall reaches the trench floor, amphipods detect dissolved chemical cues and converge from many directions. Rapid aggregation is a key trait in a habitat where large meals are rare and widely spaced. Energy from a single event is stored as wax esters and neutral lipids and used over long intervals. The sensory system is tuned for low light and for heavy reliance on chemical gradients. Amphipods at the trench floor carry numerous sensilla on antennae and mouthparts. These hair-like structures are plugged into dense arrays of neurons. The lateral body plates show pores that likely house pressure-sensitive receptors. The result is efficient detection of flow and of concentration changes. Orientation behavior has been measured in lander experiments that release tracer compounds at controlled rates. Individuals show directed movement up the concentration gradient and remain near the source until satiated or displaced by larger animals. Growth and reproduction follow a schedule shaped by temperature and pressure. Amphipods grow through molts. The timing is irregular because it depends on food pulses and on the condition of the exoskeleton. Females carry eggs and juveniles in a brood pouch under the thorax. Broods are small and the eggs are large. Embryos develop slowly. Juveniles emerge ready to feed and to avoid predators. The pattern is conservative. It favors high survival of a small number of offspring instead of large broods with high mortality. Longevity is not well constrained, but isotope turnover in tissues and mark and recapture style analyses suggest lives measured in several years. Physiology reflects pressure management. Trimethylamine oxide, spoken as trimethylamine oxide to avoid confusion in audio, is present at concentrations that increase with depth. The molecule stabilizes proteins and offsets the destabilizing effect of urea and of pressure on protein folding. Cell membranes are enriched in unsaturated fatty acids that keep the bilayer flexible. Ion channels in nerve and muscle are subtly altered in amino acid sequence to preserve opening and closing kinetics when pressure compresses water and changes viscosity. Hemocyanin, the blue copper-based oxygen carrier in many crustaceans, shows variants that maintain oxygen affinity at low temperature and high pressure. These features are consistent with a design goal of ordinary function in an environment that would arrest the physiology of shallow species. Chemical contamination is recorded directly in amphipod tissues. Polychlorinated biphenyls and related persistent pollutants occur in lipid stores at concentrations that map to global production and deposition histories. Microplastics are present in the gut contents of many trench amphipods. Fibers from synthetic textiles dominate the counts. Fragments of polyethylene and polypropylene and polyester are common. The route from coast to trench is well established. Fibers and particles enter rivers and coastal waters, attach to organic matter, and sink. Amphipods ingest them with detritus. Most pass through, but some remain for days within the gut. The measurements do not show immediate toxicity at the levels observed, yet they demonstrate that no part of the ocean floor is free of human material. Stable isotope data clarify where amphipods sit in the food web. Ratios of carbon-13 to carbon-12 and of nitrogen-15 to nitrogen-14 indicate that most hadal amphipods feed 1 to 2 steps above sinking phytodetritus. The signal shifts when large carrion is available, reflecting the higher trophic position of the food fall. Lipid biomarkers agree with the isotope story. The presence of certain long-chain fatty acids points to inputs from diatoms and from bacterial reworking of organic matter during descent. The combination of markers allows researchers to reconstruct diet over weeks rather than over hours. Locomotion at pressure requires efficient muscle. Electron microscopy of muscle fibers shows dense arrays of mitochondria and long sarcomeres that favor slow sustained contractions over rapid bursts. This matches behavior seen in video. Most movement is deliberate. Quick escape swimming is rare and brief. The emphasis is on endurance and on posture control around bait and in currents near the seafloor. The body shape reduces drag when the animal hugs the ground. When swimming is necessary, pleopods beat in coordinated series to produce steady thrust. Population structure changes with depth and with location along the trench axis. Surveys show that different species replace one another over a span of a few hundred meters in depth. This zonation likely reflects physiological limits and food availability. Temperature gradients of 1 or 2 degrees Celsius between deep basins and adjacent slopes also play a role. Genetic data reveal limited gene flow between basins that are separated by sills and by long distances. The trench is not a continuous habitat in a biological sense. It is a chain of basins and slopes with distinct conditions. Mechanical interaction with baited landers has supplied measurements that would be difficult to collect otherwise. The force with which an amphipod can grasp and manipulate bait has been estimated by the displacement of tethered weights and by strain sensors on bait arms. Values scale with body length and match the need to tear soft tissue and to carry fragments away from competition. The loads are modest in absolute terms, but they are high relative to body mass. This is consistent with the muscular investment seen in anatomy. Some hadal amphipods show gut microbes that assist digestion of refractory materials. Metagenomic surveys of gut contents identify genes that code for enzymes such as endo-beta glucanases and xylanases. These enzymes act on plant cell walls. The presence of these genes supports the observation that wood and plant debris are significant occasional foods. It also extends the role of amphipods from scavengers of animal tissue to general processors of carbon that falls to depth. Mortality events are rare in observation because the habitat is large and landers sample small areas for short times. Indirect evidence comes from the dynamics of population size structure. When a series of deployments over a season shows a surge in juveniles, a previous reproductive event is inferred. When size classes shift without recruitment, predation or export by currents may be responsible. Larger predators at hadal depths are few. Fish are the main consumers of amphipods, along with other amphipods. Cannibalism is common during crowding at bait. The amphipod chapter leads into the wider community of invertebrates. Sea cucumbers are among the most frequent large animals on abyssal plains. In the trench, they persist in smaller forms and with different body plans that handle steep slopes and soft mud. These animals, members of the echinoderm group, move slowly across the sediment using tube feet that are powered by a water vascular system. The body wall is supported by microscopic ossicles rather than by heavy plates. This choice of materials fits the chemistry of deep water where carbonate tends to dissolve. The feeding strategy is simple. A ring of tentacles collects surface sediment and transfers it to the mouth. The gut extracts bacteria and organic molecules. The remainder is egested as clean pellets that alter the microtopography. Species diversity is not high compared with the shallower abyssal plains, but functional diversity is sufficient to process available detritus. Some hadal holothurians are nearly transparent. High-resolution footage reveals the intestine and the gonads through the body wall. Others are dark and covered with short papillae that increase surface area for chemical sensing. Individuals leave tracks that persist for hours in low current conditions. Track density gives a minimum count of animals that passed within a time window. Combined with video, these traces supply estimates of activity budgets. Sea cucumbers manage pressure without the copper-based blood pigments seen in crustaceans. Oxygen transport relies on diffusion across moist surfaces and on respiratory proteins dissolved in body fluids. Metabolic rates are low. The animals spend most of their time in a slow forward crawl punctuated by pauses. When a pulse of fresh organic material arrives, measured as a change in fluorescence in the top millimeter of sediment, feeding rates increase. This flexible response is essential in a system where supply is variable. Reproduction among hadal holothurians takes two forms. Some broadcast spawn, releasing eggs and sperm into the water column. Others brood young. Brooding is common where currents are strong and where the probability that sperm and eggs meet is low. In brooding species, the adult carries embryos in a pouch or attached to the body surface until they are competent to feed on their own. Oocyte size tends to be large. Fecundity is low. The pattern mirrors the Amphipod strategy of investing in survival of fewer young. Other invertebrates share the sediment. Polychaete worms live in burrows and tubes. Their jaws are reduced when diet is dominated by bacteria and detritus. Where more active predation is possible, jaw structures are more complex. Tube builders use mucus and small particles to construct shelters that withstand the weight of overlying mud. When a turbidity current passes, many tubes are destroyed and the community resets. Recovery is measurable over months as new tubes appear and as tracks and burrow openings repopulate the surface. Isopods occupy a similar niche to amphipods but move more slowly and often burrow. Hadal isopods are less conspicuous in videos because they do not swarm to bait. They contribute to sediment mixing and to the slow turnover of organic matter. Some species show extreme sexual dimorphism, with males much smaller than females and specialized for locating mates through chemical signals. Bivalve mollusks occur near seep locations where reduced chemicals like hydrogen sulfide and methane reach the sediment surface. The bivalves host bacterial symbionts in their gills. The bacteria oxidize the reduced compounds and feed the host. These communities are patchy and small in the trench compared with continental margin seeps, but they demonstrate that chemosynthesis operates to some extent even in the deepest basins. Cnidarians are present as small anemones attached to hard fragments and to shells. Tentacles extend to collect suspended particles. The animals withstand burial by extending through thin layers of new sediment. Their distribution correlates with the presence of coarser grains where attachment is possible. On soft mud with no fragments, anemones are rare. Echinoderms other than sea cucumbers are less common but not absent. Small brittle stars occur on slopes where currents keep the surface free of fluff. They lift arms into the flow to capture particles. Sea stars adapted to soft sediment hunting are recorded less often. The scarcity of hard prey and the dominance of fine organic matter make deposit feeding a better strategy in most areas of the trench. A diverse suite of meiofauna lives between grains. Nematodes dominate counts in many samples. Harpacticoid copepods and small foraminifera share the habitat. These animals are the first consumers of bacteria and help transfer energy upward. They also stabilize the sediment surface and affect how water and solutes move across the boundary. Counts vary with the amount of organic carbon in the top centimeter. Where a recent fall of material has occurred, densities rise over weeks and then decline as the pulse is processed. Jelly-like animals are recorded in the water column close to the bottom. Small medusae and ctenophores drift past cameras and do not interact with the sediment. Their presence indicates that not all energy at depth arrives from above as particles. Some arrives as living plankton that has descended. These forms are fragile and rarely captured, so optical methods provide most observations. Crustaceans beyond amphipods include tannaids and cumatians that move in short runs and use claws to dig. Their life cycles are short compared with amphipods. They breed within a season when food is ample and retreat to low activity when it is scarce. These groups are undersampled because they avoid bait and are small enough to slip through coarse sieves used in early work. With fine mesh and with suction samplers on landers, their true abundance has become clear. Sponges are rare at hadal depths due to the lack of stable hard substrate and to low silica availability, but anchored forms do occur on occasional rock outcrops. Microbial symbionts in sponge tissues perform nitrification and other steps in the nitrogen cycle. The contribution is local but measurable in incubations that enclose a patch of seafloor. The role of invertebrates in bioturbation is central. Tracks, burrows, and feeding pits pump oxygenated water into the upper millimeters of sediment and bring reduced compounds to the surface where microbes can oxidize them. This vertical mixing increases the effective thickness of the active layer where carbon processing occurs. Rates measured with tracer techniques show that without animal activity, the oxygen penetration depth would be smaller and the efficiency of organic matter degradation would decline. In a low-energy environment, small increments matter. Predation among invertebrates is modest but present. Polychaetes with irreversible jaws prey on smaller worms and on meiofauna. Large amphipods eat smaller ones. Sea cucumbers host parasitic copepods and are occasionally attacked by nematodes that enter through the anus and feed on internal tissues. The balance of these interactions changes with food input. After a large food fall, competition at bait increases and cannibalism rates rise. During lean periods, interference declines and animals spread out. Adaptations to steep slopes include body shapes that resist sliding and behaviors that anchor the animal. Sea cucumbers use tube feet to hold position. Polychaetes extend parapodia laterally to increase friction. Amphipods tuck under small ridges and face into the current. Landers that record for days show that animals maintain position during slow mass movements of fluff sediment triggered by small earthquakes. When failure occurs on a larger scale, the community is reset in that sector and recolonization begins from adjacent areas. Sampling at these depths requires hardware modifications, nets that close at the bottom to prevent loss of small animals during ascent, pressure chambers that keep captured organisms at in situ pressure, and lights that work at low intensity to avoid attraction or avoidance responses are now standard. These tools have reduced the bias that once favored large scavengers in datasets. The result is a more balanced view of the invertebrate assemblage. Interactions between macrofauna and microbes are two-way. Sea cucumbers and polychaetes irrigate sediment and supply oxygen that bacteria use. Bacteria produce extracellular polymers that stabilize fecal pellets and make them easier for deposit feeders to handle. Amphipods carry microbial communities on their exoskeletons that process dissolved organic matter in the boundary layer as the animals move. This feedback improves capture of resources that would otherwise be lost. Energy budgets derived from respirometry and from activity monitoring show narrow margins. For many invertebrates at the trench floor, annual energy intake barely covers basal metabolism and reproduction. Growth occurs in brief intervals after food pulses. The system is steady not because the input is constant, but because the community has evolved to match variable input with flexible demand. The giant amphipods and the sea cucumbers are therefore not curiosities. They are the principal workers that keep carbon moving at the deepest point. Amphipods locate, shred, and redistribute food. Sea cucumbers and other deposit feeders process the sediment blanket that accumulates when pulses end. Worms and small crustaceans mix the surface layer and maintain exchange with the water above. The structure is simple and efficient. It functions year after year under pressure and cold that are outside the design limits of most organisms on Earth. The transition from amphipods to holothurians and then to the rest of the invertebrate guilds shows a consistent theme. The trench is not empty. It is organized around a limited energy supply and around physical rules that make some materials and strategies more viable than others. Carbonate is discouraged, so skeletons are light or replaced by other chemistries. Rapid growth is discouraged, so life cycles stretch and reproduction invests in fewer young. Active pursuit is discouraged, so feeding is based on detritus and on scavenging. Within those constraints, the variety of forms and behaviors is sufficient to occupy the space and to keep the system stable. With these two chapters in place, the picture of the deepest ecosystem gains resolution. The small and the large are linked by chemistry and by the physics of sinking particles. The camera frames show more than silhouettes. They show a working seafloor where each taxon holds a narrow function that supports the rest. The data behind the images confirm that this function is durable across seasons and across years. The message is consistent. The deepest site on the planet is not a limit that excludes. It is a filter that selects for designs that match pressure, cold, and low energy. Pollution is recorded here with the same precision that biology is recorded. The deepest sediments and the animals that live within them hold chemicals that were made for factories and for household use many decades ago. Polychlorinated biphenyls, the group of compounds once used as coolants and in electrical equipment, appear in extracts from amphipod tissues and from mud cores. They are joined by other persistent organic pollutants that were designed not to break down easily. These molecules are hydrophobic. They attach to tiny particles and to organic flakes as they sink. The trench is a collector of such particles because its V-shaped walls and focused currents act as a funnel. Over years and over decades, the slow rain of matter carries a history of the surface into the Hadal zone. Microplastics provide the most visible evidence, fibers and fragments that began as synthetic clothing or as packaging drift down after binding to organic material. In the laboratory, filtration of sediment slurries, followed by Raman spectroscopy, identifies polymer types such as polyethylene, polypropylene, and polyester. Counts of fibers per gram of dry sediment are not zero at any depth tested. Amphipods and other deposit feeders ingest these particles along with detritus. Most fragments pass through the gut within hours or days. Some remain longer and can be found embedded in mucus within the intestines. The quantities measured do not show a single dramatic number. They show a steady presence that mirrors the ubiquity of plastic at the surface. A separate signal comes from radiocarbon, the isotope carbon-14. During the 1950s and the 1960s, atmospheric nuclear tests increased the ratio of carbon-14 in surface waters. That tracer, sometimes called bomb carbon, has worked its way through the ocean. In trench amphipods, the radiocarbon content of some tissues is slightly elevated compared with what would be expected if the animals fed only on very old carbon. The conclusion is that at least part of their diet is recent, with a surface origin. That is consistent with video records that show these animals swarming to pieces of fresh carrion and to wood. Heavy metals add to the picture. The metal element mercury and the metal element lead occur in small concentrations in trench sediments. The metal element cadmium and the element arsenic are also present at low levels. These metals arrive attached to particles and remain locked in minerals or in organic complexes. In animals, the levels are generally low because the food web is short and dilution through growth is large. Even so, the presence of these elements confirms that no current moves without carrying some trace of the continents and of human industry. Antibiotic residues are a subtle case. Direct measurements are few because concentrations fall below the detection limits of many methods. Indirect measurements look for genes that encode enzymes capable of degrading antibiotic molecules. These genes appear in metagenomes from trench sediments. This does not prove contamination by modern pharmaceuticals. Resistance genes are ancient and occur naturally. The sober interpretation is that microbial communities in the trench carry a diverse toolkit, and that some of those tools happen to target chemicals that humans also target. Wider chemistry in the sediment shows how the system processes what arrives. Lipid biomarkers track the origin of organic matter. Long-chain alkanones indicate inputs from haptophyte algae. Specific sterols point to diatoms. Pigments are degraded, but their breakdown products still speak of surface blooms. As the material descends, it is reworked by microbes. Proteins are cleaved to amino acids and then to ammonia. Carbohydrates are hydrolyzed to simple sugars and then oxidized. What reaches the trench floor is not pristine. It is a selected mixture that still holds enough energy to support a community. Alongside these natural compounds ride the synthetic molecules that tell us the deep sea is connected to every river mouth and every coastal city. The record of pollution has one more element that is not chemical. In the late 2010s, video from a deep lander showed a piece of plastic film on the trench floor. The object did not affect any measured process. It lay on the mud as any other piece of debris would lie. Its importance was symbolic. It made plain in a single frame what datasets had been implying for years. If a bag can reach this place, so can fibers, microbeads, and unwanted additives. The conclusion is not an appeal. It is a fact that sets the context for the biology described here. The discovery of new species in this environment proceeds slowly and without publicity. Names are assigned only after careful anatomical and genetic work. Among the crustaceans, the Amphipod genera that dominate baited lander videos include species that were unknown 2 decades ago. One now well-known example is the Amphipod Eurythanes plasticus, pronounced Uri, then Ees plas T-cus, described from another trench. Its name acknowledges the presence of plastic in its gut, but the species is defined by anatomy and by gene sequences. In the Mariana region, surveys continue to reveal members of the same genus that differ in size, in the shape of the gnathopods, and in the morphology of mouthparts. Each difference maps to a gene tree that shows separation across basins and across depth ranges. Beyond amphipods, ctenidaceans and cumaceans present a steady stream of new records. These small crustaceans burrow and avoid cameras. They enter samples when corals recover intact surface layers or when suction samplers sweep the top few centimeters of mud into a chamber. Species descriptions note the shape of the carapace, the number and arrangement of setae on appendages, and the form of uropods. Many are endemic to single trenches, or even to segments of a trench. Their life cycles appear short and flexible, suited to exploiting brief pulses of food. Isopods at hadal depths include lineages with extreme sexual dimorphism. Males are tiny compared with females and lack functional mouthparts in some species. Their lives are focused on locating a female and fertilizing eggs. Females feed on detritus and on microbial mats on the surface of the sediment. Such strategies reduce competition between sexes and guarantee that sparse populations can still reproduce. Descriptions of these isopods rely on delicate features that are easily damaged during ascent. Pressure-retaining containers and gentle sieving have improved the quality of specimens and the clarity of the taxonomy. Among worms, the polychaetes provide the richest set of new species. Deposit-feeding families such as capitellids occur widely. Tube-building amphorettids and terebellids construct small dwellings from mucus and silt and extend palps across the sediment to gather particles. Predator families such as psyllids and dorvillids have irreversible jaws that grip small prey. The deep environment selects for reduced calcification. Setae and jaws are robust where needed, but the rest of the body is light. Many trench polychaetes show enlarged parapodia that help them stay in place on slopes. Their burrows and tubes increase the exchange of water within the top layer of sediment, and that, in turn, increases the activity of bacteria. A peculiar group of worms called pogonophorans, also known as beardworms, occur near seeps in the trench region. They lack a digestive tract as adults and rely on endosymbiotic bacteria for nutrition. The bacteria oxidize reduced chemicals such as sulfide or methane when those molecules are present in the pore waters. The worms supply oxygen and other substrates through a vascular system. Together, they form a living reaction network that converts geochemical energy into biomass with high efficiency. New species of these worms continue to be described from slope sites adjacent to the main trench axis. Nematodes dominate the meiofauna. Their diversity is high, and many lineages are known only from molecular barcodes at present. In some cores, the number of nematode species rivals counts from shallower depths, although the abundances per square centimeter are lower. The small body size allows nematodes to exploit microenvironments created by grains and by biological structures. Their feeding modes range from bacterial grazing to predation on other nematodes. Each new sequence added to genetic databases becomes an anchor for future studies that will connect names to functions. Annelids beyond polychaetes include rare oligochaetes that resemble earthworms in basic design but are adapted to saline water. They move through sediment slowly and process organic matter. Kinorhinchs, sometimes called mud dragons because of their spiny plates and their segmented appearance, also occur in small numbers. They grasp grains with well-developed mouth parts and pull themselves forward with a telescoping motion. For many of these small taxa, the trench represents the limit of their distribution, not because of pressure alone, but because the energy available per square meter is low. New species survive here by minimizing demand and by maximizing flexibility in diet. To connect these biological discoveries with the geology of the region, one must address fluids that percolate upward through the crust. Hydrothermal vents, in the strict sense of very hot springs that precipitate metal sulfides and form chimneys, are most common along mid-ocean ridges and in back arc basins. The axis of the Mariana Trench is not such a place. However, the broader subduction zone includes mud volcanoes and seeps on the forearc, where fluids altered by reactions in the subducting slab rise to the seafloor. These fluids are alkaline and cool to warm rather than hot. They carry dissolved hydrogen, methane, and small amounts of sulfide. They create localized chemistry that differs sharply from ordinary deep water. At some seamounts near the trench, thick flows of serpentinized mud ooze from vents and shape low domes. The temperatures measured in the fluids are only a few degrees above ambient seawater. The alkalinity is high. The pH, which we will call the alkalinity level to avoid abbreviations, reaches values near 12 in some samples. In these places, carbonate minerals precipitate as unique forms. Brucite and aragonite form chimneys that are fragile and pale. Within these structures, microbial communities thrive on hydrogen and on methane. They use enzymes that split hydrogen to capture energy and enzymes that oxidize methane. Bivalves and gastropods host symbiotic bacteria that process these reduced chemicals. The animals live as thin communities scattered across vents where flow is steady. On slopes within the trench, low-level seeps exhale fluids that are less alkaline but still enriched in reduced compounds. Here, mats of filamentous bacteria grow, producing sulfur granules inside their cells. Small polychaetes graze on the mats. Bivalves with symbionts colonize patches where sulfide flux is persistent. These seep communities are small compared with famous ridge vents, but they demonstrate that the subduction environment feeds chemosynthesis in more than one way. The result is that carbon entering the trench from surface production is not the only source of energy. Crustal chemistry contributes a second path. Mineral evidence of past hydrothermal activity appears in sediment records as layers enriched in barium and in silica. These signals likely mark periods when plume fallout from back arc spreading centers reached the trench region. Volcanic ash layers also occur. Each layer is a time marker. Dating them with radiometric methods allows correlation with known eruptions and with known episodes of increased hydrothermal output. Biological markers sometimes coincide, as when the diversity of siliceous sponges or radiolarians changes in bands that match the chemistry. Hydrothermal and seep fluids alter the mechanical properties of sediment. poor waters with high alkalinity levels can cement grains and make crusts that resist erosion. When such crusts form on steep slopes, they can stabilize patches of habitat and allow colonization by animals that need a firm base. In other places, gas bubbles accumulate and weaken layers. Small failures occur and leave scars visible in side-scan sonar. These events reshape the micro-topography that in turn influences where invertebrates settle and feed. The microbiology of vent and seep sites near the trench shows metabolic versatility. Hydrogen oxidizing bacteria use a group of enzymes called hydrogenases to capture electrons from hydrogen and to feed them into energy pathways. Methane oxidizing bacteria use particulate methane monooxygenase and soluble methane monooxygenase enzymes to convert methane into methanol and then into carbon dioxide while harvesting energy. Sulfur oxidizing bacteria run pathways that convert sulfide to sulfate. In combination, these processes support a food web that is independent of sunlight. Many strains show pressure-adapted versions of their enzymes, with amino acid substitutions that stabilize active sites in compressed water. Membrane lipids are more unsaturated to maintain fluidity. These are the same themes seen in non-chemosynthetic communities tuned to a different energy source. The physics of seepage sets a slow rhythm. Subducting plates dewater as they descend. The released fluids percolate through faults and through permeable layers. Flow rates at the seafloor are low by human standards, measured in centimeters per day to meters per day. Even so, the gradients created are enough to sustain communities for decades. When flow shifts, communities decline and a new patch comes to life elsewhere. This mosaic is one reason why surveys over many years are required to build a map of chemosynthetic habitats in the trench region. Chemical signatures of these fluids appear in the stable isotope composition of carbon in animal tissues. Values shifted toward very light carbon indicate a diet based on methane. Values closer to the marine plankton average indicate reliance on photosynthetically derived material. Animals can switch between sources. Bivalves with symbionts may show a strong methane signal, while their neighbors show a mixed diet. The plasticity supports survival in a patchy environment. The geology also affects oxygen levels in poor waters. Where reduced fluids rise, oxygen is consumed quickly. Redox boundaries sharpen. Microbial diversity is high at these boundaries because many processes can proceed side by side. Iron reducers and manganese reducers occupy thin bands adjacent to oxidizers. Denitrifiers work where nitrate diffuses downward from the water column. The resulting layered chemistry creates habitats that support meiofauna with specific preferences. Nematodes select particular depths. Foraminifera choose depths where their symbionts can find the right balance of oxidants and reductants. A word about temperature helps keep expectations realistic. The Hadal environment close to the trench axis is cold. With the exception of local seeps and mud volcanoes, there are no hotspots. Black smoker chimneys do not rise from the trench floor. The existence of chemosynthetic communities here relies on the chemistry of loss rather than on the chemistry of heat. Serpentinization of ultramafic rocks produces hydrogen. Decomposition of subducted organic matter produces methane. The sums are small compared with ridge vents, but they are sufficient to sustain life that specializes in low flux. As with other parts of the trench story, technology determines resolution, chemical sensors capable of detecting nanomolar changes in sulfide and in methane guide cameras to patches of activity. Mass spectrometers operate in situ and report compositions in real time. Flow meters record slow exhalations and inhalations of the seafloor. Pressure-retaining samplers recover fluids without degassing and permit laboratory analysis of dissolved gases and of microbial cells. Each instrument adds clarity to a picture that had been guessed at for years. The three themes of this chapter form a single system. Pollution gives a record of connection to the surface. New species of crustaceans and worms show how life adapts to the most constrained budgets and to the most stable of environments. Hydrothermal and seepage processes provide a second stream of energy that intersects with the first. Together they explain why the deepest place on Earth is not barren. It is structured, busy at a slow pace, and sensitive to inputs from both above and below. Mineral formations at the trench floor record chemistry with the discipline of a logbook. The first rule is the scarcity of calcium carbonate. In the Pacific, the level at which calcium carbonate dissolves sits far above the trench axis. Below that level, shells of calcite and of aragonite tend to disappear. What arrives from the surface is dissolved and redistributed before it can build a lasting layer. As a result, the sediment is dominated by clay, by volcanic ash altered to new minerals, and by particles that form directly within the mud through slow reactions. Where carbonates do occur, they are usually borne in place as products of unusual local chemistry rather than as rain from above. One such local chemistry appears near alkaline seeps on the forearc. Fluids rich in dissolved carbonate species precipitate fragile forms of aragonite and brucite as they meet ordinary seawater. The result is a foam of white chimneys and crusts that are light enough to crumble at a touch. Samples show delicate needles and rosettes when examined under a microscope. The crystals grow grain by grain as flow continues. When the flow stops, the structures become fossils of chemistry rather than living conduits. Away from those seep patches, the trench sediments show other orthogenic minerals. Barite, a mineral of barium sulfate, forms tiny rosettes where sulfate reduction and reoxidation create microenvironments that favor its precipitation. The rosettes persist as beads in the clay. Phosphate minerals occur where organic matter has been buried and degraded. In thin layers, they cement grains into subtle hard grounds that resist erosion and change how animals can feed. Pyrite appears as framboids, the classic clusters of tiny crystals that resemble raspberries. These framboids mark zones where microbes have used sulfate as an electron acceptor and created sulfide that then reacted with dissolved iron. Silica records add another line to the mineral chapter. Skeletons of radiolarians and of diatoms fall from above and deliver opaline silica. Most dissolve during descent or within the upper sediment, but some persist and become part of a mixed matrix that later flows downslope as turbidites. In places where volcanic ash layers were delivered by eruptions from arc volcanoes, that ash alters slowly to zeolites such as phillipsite and clinoptilolite. These alteration minerals trap cations and water within frameworks that tell geologists about the time the ash spent within the seawater column and within pore waters. In cores, The presence of zeolites marks intervals of ash input and helps identify sources when chemistry is compared to known volcanoes. Hard rock is scarce on the trench floor because sedimentation is active and slopes are steep, but where outcrops exist, they carry their own mineral record. Iron and manganese crusts coat the rock with a thin, dark film. These ferromanganese crusts grow at rates of millimeters per million years. They are hydrogenous in origin, accumulating directly from metal ions dissolved in seawater. Their chemical layers hold traces of rare metals and of isotope ratios that reflect changes in the ocean over long spans. A chipped fragment of such a crust can therefore act as a small history book of marine chemistry. Grains within the trench sediment sometimes bear a varnish of manganese oxides that also accumulates from seawater. The varnish darkens the grains and increases their density. Under a scanning instrument, tiny platelets of bernacite and other manganese oxides appear, layered with iron oxyhydroxides. The balance of iron and manganese can shift with microenvironmental redox changes, which means the varnish is both a product and a record of the chemistry of the thin film of water that moves through the sediment bed. Volcanic contributions are frequent because the trench parallels a chain of islands with active and dormant cones. Ash falls enter the ocean and settle through the water column. In cores taken near the trench axis, distinct ash layers alternate with clay. The glass shards that make up the ash alter to pelagonite and then to zeolites through slow reactions with seawater. Trace element signatures, such as ratios of strontium to neodymium and of rare earth elements, allow correlation with eruptions on land. In this way, a quiet mud core contains time-stamped pages written by distant volcanoes. Not all mineral features grow quietly. Some are created by rapid deposition during slope failures. When currents rush downslope after an earthquake, they carry a mixture of silt, sand, and fragments that settles as a graded bed, coarser at the base and finer at the top. Within these beds, oriented mica flakes and imbricated clasts record flow direction. The base of a large event bed can cut down into older layers, leaving an erosional surface that appears in seismic profiles as a reflector. Cementation within these deposits can create lenses that later stand out as firmer patches compared with surrounding mud. Ancient processes leave macroscopic and microscopic signatures that let geologists reconstruct events step by step. Turbidites appear as repeated sequences of coarse to fine layers, the boomer sequence familiar to sedimentologists. Between these event beds lie intervals of slow pelagic rain, the red clay that builds grain by grain over thousands of years. Tephra layers from eruptions are thin and widespread. They are dated by radiometric methods and by paleomagnetism. The reversal of the magnetic field known as the Matuyama to Brunhes boundary, which occurred roughly 750,000 years ago, appears as a flip in the orientation of magnetic grains within cores. That flip anchors the age model for many sections. At rarer intervals, microtectites appear in deep ocean cores. These glassy spherules were formed when meteorites struck continents or shallow seas and melted target rocks that were then launched and cooled in the air. While not abundant within the trench axis where deposition is complex, they occur in nearby deep basins and provide correlation points for impacts, including the event that marked the boundary between the Cretaceous period and the Paleogene. Their presence in Pacific records shows how far such debris can travel and how faithfully the ocean keeps the pieces. Mass wasting leaves scars on the seafloor that modern mapping can see in detail. High-resolution multi-beam surveys show step-like features called scarps, hummocky terrain below slide sources, and long run-out deposits where flows pond within basins. Some scars align with faults created as the oceanic plate bends into the trench. Those bending faults cut the crust and provide pathways for fluids. Where water percolates along them, alteration of olivine and pyroxene to serpentine occurs, a reaction that produces hydrogen and changes rock density. The topographic record of such processes is subtle but detectable as ridges and troughs that parallel the trench. Terraces along the inner wall of the trench tell of repeated slumping and of slow backstop movement as material is scraped off the subducting plate and added to the upper plate as an accretionary prism. Each terrace holds a stack of deformed layers, folded and faulted. Seismic lines show packages of reflectors that dip landward, the fingerprint of accretion. Between terraces, canyons carry sediment down to the axis. Their heads are often linked to rivers on the islands through a chain of gullies and shelf breaks, demonstrating a full pathway from volcanic flank to trench floor. Some cores contain layers enriched in the element iridium and in chrome-rich spinels, signatures associated with meteorite material. These layers are thin and require careful chemical separation to detect. Where found, they point to local fallout from distant impacts or from reworking of older impact layers by currents. While not headline features, such layers add to the quiet evidence that global events leave marks even in the most remote basins. Gas hydrates are a potential mineral phase where pressure is high and temperature is low. These ice-like lattices that trap methane and other gases are common on some continental margins at depths much shallower than the trench. Within the trench axis, direct sampling of hydrate is scarce, likely because organic matter delivery is limited and because the geology does not favor the formation of thick hydrate layers. On adjacent slopes and in the forearc, hydrate indicators appear more often. Seismic blanking and pockmarks suggest past episodes of gas escape. The implication is practical. The trench is not a reservoir for hydrates, but the broader subduction environment cycles gas that can from time to time form hydrate in favorable pockets. A separate set of markers of ancient processes resides in the clay mineral assemblage. The relative abundance of elite kaolinite, smectite, and chlorite reflects the balance between volcanic inputs, continental weathering products carried by wind, and in situ alteration. Shifts in those proportions track climate change on land and ocean circulation changes. For example, increased smectite can indicate more volcanic ash or more alteration of basaltic glass, while increased elite may signal stronger input from continental dust. These proxies operate slowly and provide context to the faster stories told by turbidites and tephra. Taken together, the mineral and stratigraphic record under the trench floor is not blank. It is a layered archive of steady rain, punctuated delivery from slopes, and pulses from volcanoes and from rarer global events. Each component is independent in origin but stored together. When analyzed, the combined record describes a dynamic system that concentrates, sorts, and preserves. Against this background of minerals and structures, animals that look spectral to a human eye stand out. The phrase ghost crab has been used in popular accounts to describe pale decapods at great depth. Strictly speaking, true shore ghost crabs live in shallow sands and do not occur in trenches. In the Hadal zone of the Mariana region, most decapod crustaceans are scarce for physiological reasons. The magnesium content of seawater interferes with their nerve function under very high pressure, a condition sometimes called magnesium narcosis in the literature. This limitation sets a practical depth boundary for many crabs and for many shrimps. There are exceptions and workarounds. In the broader Mariana system, at vents and at seeps in the back arc and on the forearc, decapods occur that are pale with reduced eyes and thickened setae. Some are squat lobsters of the genus Munidopsis, which is an anomuran group rather than a true crab. They carry long claws and a compact body and often look ghostly white in camera light. Others are vent crabs in families that specialize in chemosynthetic settings. They detect chemical cues with enhanced antennal sensilla and graze microbial mats or scavenge around vents. While these animals are not typical residents of the trench axis, their presence in the regional province contributes to the impression of a pale, spectral crustacean fauna. Wood falls and carcass falls provide another occasion for unusual crustaceans. On rare deployments, a piece of wood lowered by a lander has been colonized rapidly by white squat lobsters and by isopods with reduced pigmentation. They rasp the wood with mouthparts adapted for scraping and rely on gut symbionts to digest cellulose. The pale color is not an adaptation to avoid predators because there is almost no light to detect it. It is simply a consequence of living where pigment is not required. Other animals deserve the label strange because they alter body plans in ways that are uncommon elsewhere. Holothurians with fins can swim briefly above the bottom to relocate. When seen in profile, they resemble the body of a headless bird. Transparent sea cucumbers show internal anatomy in clarity that would normally require dissection. They inflate and deflate to adjust buoyancy within a narrow range. The slow undulation of their fringe structures looks dramatic in time-lapse, yet is measured and small in absolute speed. Single-celled giants called xenophyophores are part of this ghostly category in a different sense. They build tests by gluing together particles including sponge spicules and volcanic shards, and they reach sizes that the word single cell does not prepare a listener to expect. Under soft lighting, they resemble pale roses pressed into the mud. Their surfaces host other organisms, from tiny worms to bacteria. In essence, they act as islands of complexity that break the uniformity of the sediment plane. Anemones anchored to small stones or shell fragments lift translucent tentacles into currents barely perceptible to cameras. They close whenever new sediment settles and reopen minutes later. Ctenophores drift by like small lanterns, though the water above the bottom and are visible only in the beam of the camera. Polychaetes with irreversible jaws leave brief sketches in the mud when they erupt from burrows to seize prey. Most of the time, the only sign of their presence is a pair of small holes and a halo of disturbed grains. Among fish, the snailfish continues to be the emblem of a world near the edge of vertebrate possibility. Individuals filmed near the trench floor look almost gelatinous. Their bones are lightly mineralized, their bodies rich in osmolytes that stabilize proteins, and their heads bear little in the way of scales. They move with short undulations of the body, conserving energy. When they open their mouths over bait, the action is calm and assured. There is no wasteful strike. Video analysis of their movements shows efficient use of the tail finite element by finite element. The effect is spare rather than spectacular. Bioluminescence is present but subdued near the bottom. Many pelagic animals in the water column above the trench use light for communication or for camouflage. On the floor, flashes recorded by cameras are infrequent and generally weak. Some come from small ostracods that release light to distract predators. Some may come from bacteria that colonize organic particles. There is no display. There is only the occasional spark. The category strange life forms also includes animals that adjust strategies in non-intuitive ways. Some small crustaceans increase lipid storage to a degree that makes them nearly neutrally buoyant in cold water, allowing them to hover above the sediment and drift down onto food with minimal effort. Certain worms grow elaborate sensory head structures that let them map chemical gradients with precision finer than a millimeter. Anemones align their columns parallel to the weak near-bottom flows to reduce drag. Each of these features is a solution to the same problem: the economy of energy in a place where energy arrives in pulses and is scarce between them. Claims of large unknown predators or of exotic creatures unfamiliar to science have not stood when tested against good images and specimens. Most shapes resolve into known groups once observed clearly and catalogued with their parts. What remains to be described is not monsters but details, the number of segments on a mouthpart, the sequence of atoms in an enzyme, the exact composition of a mineral crust on a grain. These details are the coins in which knowledge here is bought. In summary, mineral formations in the trench combine orthogenic products like barite, pyrite, and zeolites with hydrogenous coatings of iron and manganese, and with occasional carbonates born of unusual alkalinity. Stratigraphy records ancient processes through turbidites, tephra, magnetic reversals, and subtle chemical layers. The so-called ghost crabs of the deep are mostly pale anomurans and vent decapods that dwell away from the axis, while at the axis itself, the spectral cast is supplied by pale amphipods, transparent holothurians, xenophyophores, and finely adapted worms. The system is consistent across these scales. It writes its history slowly, holds the products of chemistry carefully, and supports life that is spare and efficient.